Okay, welcome back. Now, because we said DNA does not exist as a single strand, it exists in form of a double strand. Now, how does this double strand is formed? At least we have seen how the nucleotides come together to give a dinucleotide, another one attached to form a tri, another one attached to form tetra like this, until when we have a very long nucleotide we call polynucleotide. Now, that's what we call complementary base pairing, where one nucleotide habitually form a bond with another one or complement another one. And here we say thiamine complement adenine. So wherever you have a nucleotide that contains thiamine, in this case, we are talking about the oxythymidylate. You have the oxythymidylate bind to the oxyadenylate, where the thiamine, which is the base, in this deoxythymidylate, bind to the adenine, which is a base in deoxyadenylate. And how this bond is formed is what we explained. That two hydrogen bonds help in linking the two nucleotides. While in the case of cytosine, cytosine form a bond with guanine, a complementary bond with guanine. And this bond is through 3 hydrogen bonds. And we have understood that this interaction between cytosine, or this bond between cytosine and guanine, because it is 3 hydrogen bond compared to the bond between thiamine and adenine, which is 2 hydrogen bond, it is a single indication that the interaction between cytosine and guanine is stronger than between thiamine and adenine. Is that clear? Is that clear?
Yes, sir.
Okay. Okay, now coming back to this, we have already explained this as a polynucleotide. Now this is just one strand. How does the second strand now come to complement? Now here we have another strand. This strand will now contain a nucleotide that must complement these ones. So if this one have adenine here or adenylate, what nucleotide would complement adenylate? Which nucleotide will complement adenylate? Class.
Is it time?
What?
Thiamine.
Yes. Thiamine bind to adenine. While now, what nucleotide will complement cytidylate or deoxycytidylate? What nucleotide?
Oh, yes.
Please talk. Which nucleotide will complement cytidylate? Now let me repeat. Let me repeat. We talked about this complementary base pairing, and I said in complementary base pairing, thiamine bind to adenine through 2 hydrogen bonds, while cytosine bind to guanine. So in this case, what's supposed to come to your mind is thiamine is just a base, a nitrogenous base. So also adenine is just a nitrogenous base. When a thiamine is found in a nucleotide. The nucleotide is the one that is found in DNA. We know that that nucleotide contains a deoxy sugar. Therefore, it is going to be deoxythymidylate. So wherever you see a nucleotide containing thiamine in DNA, it is deoxythymidylate, of course. So wherever you see deoxythymidylate, will bind, will complement deoxyadenylate. Deoxythymidylate will complement deoxyadenylate. Or you see deoxyadenylate, it will bind to deoxythymidylate. Whereas, wherever you see deoxyguanylate, it will bind to deoxycytidylate. Or you see deoxycytidylate, it will bind to deoxyguanylate. Is that clear?
Yes, sir.
Is that clear?
Yes, sir.
Okay, now let me use this structure. Now, we have this as a polynucleotide. If you are going to have a complementary pairing here, this one is deoxyadenylate, right? This one, this nucleotide is deoxyadenylate, right? Now, what nucleotide will complement this one? Which nucleotide?
Oxythymidylate.
Deoxythymidylate, right?
Yes, sir.
Oh, sorry. The next one now is what? This one is what?
Oxycytidylate.
This is deoxycytidylate. So now, which nucleotide will bind to now this deoxycytidylate?
Oxyguanylate.
Deoxyguanylate, yes. Deoxyguanylate. And
Guanylate.
through how many hydrogen bond? Through how many hydrogen bond?
3 hydrogen bonds.
Yes, through 3 hydrogen bond. Now, the next one here is deoxyguanylate. Which nucleotide will now bind to this one?
Oxycytidylate.
Deoxycytidylate. So also, like this. The next one is either thiamine or uracil. Remember, this is just a single strand. So it can be either thiamine if it is DNA or uracil if it is RNA. Now going back to our own, where we're trying to explain the double strand. Now you have the double strand, you have these nucleotides, adenylate, or deoxyadenylate, deoxyguanylate, deoxythymidylate, deoxycytidylate. Now they form a complementary base pairing. This one complement with deoxythymidylate. And you can see, the binding is 2 hydrogen bond, and you can see 2 dots here, which indicate the 2 hydrogen bond.
Yes, sir.
This one is deoxyguanylate, which form or complement with deoxycytidylate through 3 hydrogen bond, and we can see the 3 hydrogen bonds here. This one is deoxythymidylate. It complement with deoxyadenylate through 2 hydrogen bond, and we can see the 2 dots here, which indicate the 2 hydrogen bond. So also the last one is deoxycytidylate, which complement by deoxyguanylate through 3 hydrogen bond. We can see the 3 dots, which represent the 3 hydrogen bond. Is that clear?
Yes, sir.
Yes.
Now, another thing you need to know also, looking at this, we said, looking at the first polymer here, polynucleotide, we said it has the 5' end and the 3' end. So the complementary polymer, or the polymer that will complement, or the DNA strand that will complement this one, is this one in red, right?
Yes, sir.
So usually, this one is 5' to 3'. You can see the direction here. It is indicated 5' to 3'. So this one must be in opposite direction.
3 to 5.
5 primer to 3 primer, but in opposite direction. Here we have the 5 primer containing the hydroxyl group, while here is the 3 primer end containing the hydroxyl group. Here we have the 5 primer end containing the phosphate group. Here we have the 3 primer end containing the hydroxyl group. So the two strands of the DNA, they are anti-parallel to each other. They are complementary, but they are anti-parallel to each other. Is that clear?
Yes, sir.
Now, another thing also, we also need to explain is the fact that the DNA is double strand, but the double strand does not exist in form of linear like in this B, as we just explained. They are not just like this, like this. Rather, they are in form of helical structure. They are twisted. So just imagine this structure twisted. It is twisted to form something like this. So looking at this, we now say the diameter of the DNA is about this, which is 2 nanometer, equivalent to 20 Armstrong. So when it is twisted to form a helical structure. And this is the findings we explained that was done by James Watson and Francis Crick in 1953. While talking about some findings in biochemistry, we said that James Watson and Francis Crick were the ones that discovered the DNA helical structure. And this is what they actually discovered, that this double strand does not exist in a linear form. Rather, it forms a helical structure. And this helical structure is done in such a way that 1 turn, 1 cycle comprises of about 10 nucleotide base pair, and it is about 3.4 nanometer per length, which is equivalent to 34 Armstrong. And when this twisting occurs, the DNA produces two holes or two pockets. They are called groups. The smaller one and the larger one. The smaller one is called the minor group, while the bigger one is called basically. Najatu is disturbing again. Are you with me now?
Yes, sir.
Okay. So another thing you also need to understand here is how is this arrangement happen? When you can see the nucleotides were arranged in such a way that the sugar and the phosphate group were at the outer side, while inside, at the inner side, you have the nitrogenous base. This is simply due to the fact that these nitrogenous bases are hydrophobic in nature. So they tend to be inside and interact with each other. While because the sugar, the ribose, is polar in nature, and the phosphate group, they are polar in nature, that is why they are positioned outside. So the DNA molecule is arranged in such a way that the hydrophilic ribose and the phosphate group form the backbone, while the hydrophobic bases were stacked with middle or inside, forming the core. Because they are hydrophobic in nature, they don't want to interact with water. Sorry, let me receive this call. Okay. So this is how this is arranged. It is not just arranged like that, but simply because of their nature of the two molecules. The hydrophobic nature of the nitrogenous bases made them to come together. That is why they were positioned inside, while the outside is now covered by the ribose sugar and the phosphate group. And the ribose sugar and the phosphate group form the backbone, being the fact that they are the ones that involved in the phosphodiester bond. Another thing also you need to know is these two strands in the DNA, because we say DNA carries the genetic information. It is one of the strands that carry the genetic information. And that strand that carries the genetic information is called the template strand, or sense strand, or coding strand. While the other strand that is not the one that carry the genetic information is called antisense strand or non-coding strand. Is that clear?
No, sir.
Is that clear?
Take again, sir.
No, sir.
The two strands in the DNA. The two strands in the DNA, because we say DNA contains genetic information. One of the strands contains the genetic information, while another is just a complement of that. So the one that contains the genetic information is called the template strand, or a sense strand, or a coding strand, while the one that does not contain the genetic is just a complementary strand. It is called antisense strand or non-coding strand. Is that clear?
Yes, sir.
Now, before I talk about this, I think I need to, well, it seems I didn't even put the slide on that. Okay, before I talk about, there's supposed to be a slide where we talk about DNA denaturation. Now, we said DNA exists in double helix. And this binding of one DNA strand to another is called hybridization. But DNA can undergo a process we call denaturation. And what is this denaturation is? You know, DNA is double and helical structurally. What is meant by denaturation is unwinding of the helical conformation. Secondly, also, dissociation of the two strands, meaning breaking of this hydrogen bond that exists between the nitrogenous base. When the helical structure is lost, the unwinding or the helical structure is now become unwind. Thereby, the structure of the DNA now become a linear like this. And the second thing is this double strand now lose to become a single strand. And how does this double strand now become a single strand? It's through breakage of this hydrogen bond that exists between the nitrogenous bases. This hydrogen bonds, which is a double hydrogen bond in the case of adenine and thymine, while triple hydrogen bond in the case of guanine and cytosine. Now, what are the reasons? What may cause this to happen? usually temperature, a high temperature, or pH, extreme pH, highly acidic pH, or highly alkaline pH can cause the breakage, winding of the helical structure, as well as the breakage of the two strands to form a single strand. Another thing also, ionic strength or salt concentration. A very high salt concentration can also link to that. Now, the temperature that can cause this loss in helical structure and separation of the two strands in the DNA, it is called melting temperature. It is called melting temperature. Now, the melting temperature of DNA differs depending on the content of the DNA. If you have a DNA, now listen to me, class. If you have a DNA that have so many guanine and cytosine, and another DNA with so many adenine and thymine, which one do you think will have a high temperature before the DNA will be denatured?
Sir,
Come again.
You have it two DNA. One DNA contain high guanine and cytosine, while another one, the other one contain high adenine and thymine. Which of these two DNA do you think require high temperature before it denature?
The one with side
Yes, the one with
cytosine and guanine.
yes,
Yes.
the one with high cytosine and guanine ratio. Why? Because the cytosine and guanine ratio contain the triple bond. Because the triple bond is more stronger than the double bond. Therefore, it requires high temperature before it got broken, right?
Yes, sir.
Yeah,
Yes, sir.
the explanation. Thank you. So, going to the next is how this DNA is now packaged to form what we know as chromosome. Because what we know in classical cell biology is chromosome. The chromosome contains the DNA, or it is the DNA that is packed to form a chromosome. And this chromosome is located inside the nucleus of a cell, right?
Yes.
Yeah.
Yes, sir.
Why do we even need to pack this DNA to form a chromosome? Number one, looking at the fact that this DNA we are seeing, DNA in an organism contain about a DNA in a particular organism contain about 3 billion nucleotide base. Now imagine 3 billion nucleotide base exists in form of this double helical structure. How do you think this can all be packed inside one cell, inside the cell, even inside the nucleus? Because when you have this 3 billion nucleotide base in form of double helical structure, length of the double helical structure is about 2 meters. Now just imagine if you are to elongate it 2 meters long. How do you pack something that is 2 meters long into very small something like a cell? The cell that you cannot even see without a microscope. Now just imagine. So this DNA will have to be packed in such a way that it will be very tiny, that it can even enter into the nucleus. Why? How does this? That's one point based on the size. Another point is based on the fact that in DNA double helical structure, the DNA molecule is not stable. So the molecule is more stable when it is compacted into a chromosome. So in a chromosome, it tends to be more stable.
It's okay, but I have a
Or para. Or para. Juliet.
Yes, sir. Yes, sir.
Why are you disturbing the class?
Sorry, sir.
Why are you disturbing the class?
I'm sorry, sir.
Are you guys in a class?
No, sir. Sorry, sir.
Please mute yourself. Mute yourself.
Okay, sir.
Now, I mentioned the first reason is based on the fact that the DNA is very long, containing about 3 billion nitrogen nucleotide base pair. Therefore, as long as 2 meters will have to be packed if it has to be added or it has to be put in a cell. So it has to be packaged in such a way that it will become a very small so that it can easily enter into the cell. So based on the size, another thing is stability. The DNA double helical structure is not as stable as the DNA in form of chromosome, simply because in the DNA helical structure, the nucleotides are exposed and they are prone to a risk of so many oxidations. Substances can easily, so many substances in the cell, if they are to be dropped like this, they can attack and cause a lot of oxidation, a lot of damage, can introduce a lot of mutations, and that can lead a lot of issues. We know mutations is involved in cancer and so many diseases. Another point is also during cell division, it's very easy, to separate or to transmit the genetic information from parent to daughter cells when they exist in form of chromosome than when they exist in form of a normal DNA double helical structure. It is very difficult to transmit this information to the daughter cells. Now, how does this packaging take place? The packaging, we know initially we have the DNA double helical structure with 2 nanometer diameter. This helical structure is now wrapped around a protein called histones. This protein histones contain high quantity of a positively charged amino acids. Can we remember any positively charged amino acid? Can we remember any positively charged amino acid? Class. Can we remember any positively charged amino acid? Okay, positively charged amino acid.
Lysine.
Positively
Omega.
charged
Histidine and
amino acid, sorry?
Histidine and lysine.
Yes, histidine, lysine, arginine. So all these amino acids are mostly found in histone. Therefore, they give histone a positive charge. So the whole protein have a positive charge because of these amino acids that are found in abundance. So the whole protein is what? Is positively charged. And we also know that DNA is negatively charged, right? Because of what? Because of the negative charge in the phosphate group. And we know that when we have positive, and we know that like charges repelled and unlike charges attract. So this is just another approach of making this binding to be very tight between the two molecules. So you have the histone octamer, 8 of these histone molecules will now wrap, and then wrap the DNA around these two molecules. These are histone, 8 histone molecule to form what we call a nucleosome. So this nucleosome, you can see my cursor here. You can see this DNA strand has been wrapped around this 8 histone nucleotides to form something like this. And you see like this, like this, like this. And this is called nucleosome. And this nucleosome, for nucleosome, is contain 8 histone protein, making up a molecule called nucleosome that is 10 nanometer in diameter. So this nucleosome form this string, something like a bead. We know what is bead, right? We know what is a bead. This is just like a, right? This is like the bead, while this is like the string, a thread that attach the beads together. So it forms this structure like the bead. Structure of the nucleosome is also further coiled together. When it's coiled, it forms a tight helical fiber of about 30 nanometer in diameter. So this one also further is coiled to form what we call a chromatin fiber. This chromatin fiber is about 300 nanometer in diameter. So the supercoiled chromatin fiber also condensed and stacked to form a heterochromatin, which is the chromosome, which is about 700 nanometer in diameter. So the overall chromosome now is about 1,400 nanometer in diameter. So this is how this DNA strand is being folded, coiled, supercoiled, stacked and condensed to form a chromosome. Is that clear, class?
Yes, sir.
Yes, sir.
Do you have a question?
No, sir.
Okay, let me take a 2-minute break. I'm coming into.